There are three metabolic pathways that drive movement. They are all similar in the way that their endpoint is to produce the molecule adenosine triphosphate, which is the cellular currency for muscles to contract. These three pathways are different in several ways. Firstly, how quickly they can produce ATP to drive movement. Secondly, how long they can sustain generation of ATP for. And thirdly, the variation in yield of ATP that is produced. The simplest explanation of all these pathways is just the conversion of energy from a state of biochemical energy to mechanical kinetic energy, i.e. movement. The first pathway is the phosphocreatine pathway. This pathway is critical for power events, which typically last seconds, such as striking a ball with a bat, throwing a punch, weightlifting, throwing a javelin, or shot put. It's the pathway critical for immediate supply of energy for intensive contractile activities. Activities that require instantaneous large supplies of ATP due to recruitment of large number of motor units. Its role is of particular importance also at the onset of exercise and during transitions to more intense activity, before other metabolic pathways have time to respond. The second pathway is anaerobic glycolysis, or the Glucose-Pyruvate-Lactate pathway. This pathway is critical for high-intensity exercise where the metabolic demand for oxygen outstrips the supply in the tissue. It can be sustained up to around a minute of intense activity. An example of an activity where this pathway is important is sprinting. It requires ATP quickly to meet the increased demands of an intense workload. This metabolic pathway is limited to around 1 minute before performance starts to diminish due to the accumulation of lactic acid. The acid accumulates, dropping the tissue pH and inhibiting the enzymes responsible in the pathway, and therefore decreasing the overall ATP production. The reduction in ATP therefore impairs the contractile performance and contributes to the muscle fatigue. This is the reason why we can't sprint forever, and we have a finite period of time before our performance or our sprinting performance drops off. This metabolic pathway is also particularly important when commencing exercise from rest, as it acts as a metabolic bridge until the slower aerobic pathway can take over. Aerobic metabolism is the third pathway. This pathway is dependent on oxygen and is pivotal for the supply of energy during prolonged exercise, such as in endurance sports. It is the slowest of the metabolic pathways, but offers the highest ATP yield of all the pathways. Aerobic metabolism of 1 gram of glucose produces 30 ATP molecules, as compared to the anaerobic glycolysis pathway, which produces only net 2 ATP molecules. This graph demonstrates how quickly the 3 metabolic pathways can start producing ATP in response to initiation of exercise from a resting state. You can see the phosphocreatine pathway is immediate in its generation of ATP, but it quickly subsides, with the anaerobic system taking over. As the body adapts to exercise and more blood reaches the muscles, the supply of oxygen to the muscle tissue increases, allowing for aerobic metabolism to take over. As previously mentioned, the aim of all three pathways is to produce adenosine triphosphate, or ATP, to drive movement. Generally speaking, metabolism involves the breakdown of large molecules to smaller ones, releasing energy along the way. Energy-releasing biochemical reactions are coupled to energy-requiring biochemical reactions, allowing energy-intensive products to be made. All three muscle metabolic pathways, phosphocreatine, anaerobic glycolysis, and oxidative metabolism, releases energy that is used to form ATP molecules from ADP and a single phosphate ion. The ATP molecule is stored potential biochemical energy that, when broken up or hydrolyzed, releases energy that is used to produce contractions of the muscle. It essentially transfers the stored biochemical energy to mechanical energy driving movement. So how does ATP drive movement? What are the mechanisms behind ATP-induced muscle contraction? Skeletal muscle is ordered into bundles of muscle fibers called fascicles. Each muscle fiber or muscle cell contains cylindrical elements called myofibrils. In the myofibrils are end-to-end chains of repeating units called sarcomeres. The sarcomeres consist of two types of myofilaments, one thin type called actin and the other a thicker called myosin. On the myosin are head-like projections. These heads have areas where ATP binds to it. When ATP is hydrolyzed, it releases energy, moving the myosin head, ultimately leading to a sequence of events that cause a power stroke and essentially a shortening of the sarcomere. With shortening of the sarcomeres, the muscle essentially contracts. This process is called cross-bridge cycling, and the shortening of sarcomere is called sliding filament theory. On a side note, have you ever wondered what rigor mortis actually is? In death, the limbs become stiff and is characterized by a clinical term called rigor mortis. The limbs are stiff due to the myosin head permanently being stuck to the actin filaments due to the absence of ATP molecules in dead tissues. Anyway, let's get back to discussing the three main metabolic pathways that produce ATP for movement. This diagram tries to demonstrate the phosphocreatine pathway in more detail. ATP and phosphocreatine are both stored in skeletal muscle cells and are available for immediate use. Phosphocreatine storage is 3.5 times greater than ATP. The ATP present in muscle can only sustain intense contractile activity for a few seconds because ADP accumulates and inhibits muscle contraction. The release of energy from the breakdown of phosphocreatine is directly coupled with the reformation of new ATP. The breakdown of phosphocreatine releases energy that is used to reform ATP from ADP, allowing intense muscle contractions to continue. However, as more ATP is produced, more phosphocreatine stores get utilized and becomes depleted, limiting the period at which maximal intensity contractions can occur. This pathway is an immediate source of ATP and produces it at high rates during exercise, but its capacity is limited and can be only sustained for short periods of time. The pathway does not require oxygen and therefore is classed as an anaerobic pathway. Both this pathway and the anaerobic glycolysis pathway are crucial when the demand for ATP is greater than the available stores of ATP. These scenarios include the transition from rest to exercise or the transition to higher exercise intensities, as the aerobic metabolism is slow to react to increased demands. The anaerobic pathways are also crucial at high intensities of exercise that exceed VO2 max, but as previously said, can only be sustained for short periods. Understanding this pathway raises the question, can performance be improved from consuming creatine supplementation? There is evidence that oral creatine supplementation increases total creatine and phosphocreatine levels in the muscle. Increased muscle content through supplementation has also demonstrated increased muscle torque generation. Short sprints, resistance, and maximal intensity exercises have demonstrated improved performance. The greatest improvements in performance following supplementation has been in repetitive, high-intensity exercise bouts, which highlights beneficial use in athletes where repeated short sprints or ballistic movements are required. There is also evidence that supplementation in conjunction with resistance training enhances muscle hypertrophy, or in other words, increases muscle size. Timing of consumption has also been investigated. It has demonstrated that consuming creatine supplementation prior to and immediately following exercise is superior to consuming it in a twice daily regime in the morning and in the evening. With intense skeletal workload, where the metabolic demands are greater than the oxygen supply, anaerobic metabolism occurs. Muscle glycogen is rapidly broken down to glucose. Glucose is then broken down to pyruvate. This yields only 2 ATP molecules per 1 glucose molecule. Therefore, this pathway is rapid but poor yielding, and therefore muscle glycogen stores are rapidly depleted. If the tissue's anaerobic state persists, pyruvate is converted to lactic acid. The lactic acid rapidly dissociates to a hydrogen ion and lactate. The accumulation of hydrogen ions lowers the pH in the muscles. This inhibits further glycolysis by reducing the activity of the enzymes responsible for it. The associated decrease in ATP production impairs the contractile process and thereby contributes to muscle fatigue. This is the reason we can't infinitely sprint, and our metabolic parameters dictate that our performance deteriorates with time. Good sprinters have a higher proportion of fast-twitch muscle fibers. Fast-twitch muscle fibers contain greater levels of enzymes responsible for this metabolic pathway. Studying this metabolic pathway highlights the importance of low aerobic intensity warm-downs after lactic acid-producing bouts of high-intensity exercise. As you will see in the next slide, with aerobic metabolism, lactate can actually be used as a metabolite when oxygen is readily available. It can be used by the muscle, heart, and liver. Low-intensity aerobic cooldowns will therefore lower the concentration of lactate in the muscle and speed recovery after sprint sessions. If you stop straight after sprint training, it will take longer for your muscles to recover, as the lactic acid persists for longer in this tissue. When adequate supply of oxygen reaches the active muscle and the supply meets the metabolic demands of the tissue, aerobic metabolism occurs. The pathway is relatively slow to get up and running compared to the phosphocreatine and anaerobic pathways on commencing activity from rest, as the body stores only a small amount of oxygen in the blood, and the cardiovascular and respiratory systems require about 1 to 2 minutes to increase the oxygen delivery to the muscles to support oxidative metabolism. This pathway has the greatest yield of ATP molecules compared to any of the others, and therefore is the most energy efficient. The oxygen-dependent metabolic pathways are undertaken in the cellular components called mitochondria. They are the engine houses of aerobic metabolism. Carbohydrates in the form of glucose and fats in the form of fatty acids are the predominant metabolites that undergo oxidative metabolism to produce ATP. Under aerobic conditions, the muscle is also able to recycle the lactate produced during intense anaerobic periods. The oxidative metabolism of lactate is why during endurance events, athletes can step up a gear at pivotal times, upping the pace for limited periods of time, but then can recover by dropping down the pace, putting the supply-demand ratio of oxygen back into aerobic parameters. So let's look at this pathway more closely. Let's first talk about carbohydrate metabolism. During exercise, our body is stimulated to break down our carbohydrate stores by the hormones glucagon and adrenaline. Our carbohydrates are stored in our muscles and liver in the form of glycogen. For an average 80 kilogram man, 100 grams of glycogen is stored in the liver and 400 grams in the muscles. Glucagon stimulates the breakdown of glycogen to glucose in the liver, a process called glycogenolysis. This supplies the blood with glucose, which is fundamentally important for the supply of glucose to the brain. Adrenaline acts both on the liver and also the muscles to break down its glycogen stores to glucose. In the muscle, the glycogen molecules are broken down to glucose molecules that undergo a process called glycolysis, producing pyruvate. Unlike in anaerobic environments, due to the presence of readily available oxygen, the pyruvate molecule moves from the cytoplasm of the muscle cell into the mitochondria and undergoes a series of oxygen-dependent metabolic pathways, producing high yields of ATP. With intense aerobic exercise, glycogen stores can become depleted in around 60 to 90 minutes. As exercise is prolonged beyond an hour and liver glycogen stores become depleted, liver gluconeogenesis becomes increasingly more important. This is a process by which the liver produces glucose from non-carbohydrate metabolites, such as lactate, the amino acid alanine, and one of the products of fat breakdown called glycerol. During prolonged exercise, the predominant metabolite for liver gluconeogenesis is lactate, which is produced by the muscles in anaerobic phases of activity. When active muscle cells have alternating intensities of metabolic workload and are switching between anaerobic and aerobic states, the lactate produced can be recycled and converted back into pyruvate, which undergoes oxidative metabolism, producing ATP. Lactate can also be removed by the blood and used by distant skeletal muscle cells, heart tissue, and undergo oxidative metabolism there. Okay, let's now talk about fat metabolism. Fats are stored in adipose tissue in the form of triglycerides. Our body has a huge store of potential energy. An average 70 kilogram person has about 130,000 calories of potential energy. During exercise, adrenaline stimulates the breakdown of our fat stores, releasing fatty acids into the blood. That then is taken up by active muscle cells for oxidative metabolism under aerobic conditions. Its ATP yield is slightly less than carbohydrate oxidation, but both are vastly superior to the anaerobic and phosphocreatine pathways in terms of yield. Fats are also stored in the muscle cells, but to lesser amounts, and act as a source of fat metabolism when needed. From studying aerobic metabolism, you can see that carbohydrates and fats are crucial for supplying energy for exercise, especially for endurance activities. It also highlights the importance of getting your nutrition right. An important point to understand and use to your advantage is that the body uses carbohydrates and fats in varying proportions as an energy source in aerobic metabolism. Both the duration and the intensity of aerobic exercise determines the predominant substrate used for aerobic metabolism. At low to moderate aerobic exercise intensities, or you could define it as VO2 max of less than 50%, the predominant substrate used for metabolism is fats, whereas at higher aerobic exercise levels, such as VO2 max from 50 to 100%, carbohydrates are used as the predominant substrate. The longer the aerobic exercise lasts, the more fats are used as a predominant substrate for oxidative metabolism. This is because the body tries to conserve blood glucose for the brain as glycogen stores become increasingly depleted with prolonged activity. This information can be used to an individual's advantage. For example, if a person is overweight and their goal is to lose fat, prolonged aerobic exercise at low to moderate intensities will give the best results for breakdown of fat. Whereas if an athlete is about to undertake work at high aerobic intensities, eating carbohydrate-rich foods in the preparation phase to the event will optimize the liver and muscle glycogen stores, enhancing performance and decreasing the chance of glycogen depletion during the event, avoiding bonking or hitting the wall. If you're an ultramarathon runner, you will need to eat a higher proportion of fats than the average shorter distance endurance athlete. You will also need to regularly ingest the right types of carbohydrates during the event to avoid liver glycogen depletion, ensuring glucose is always available for the brain.